and this presentation will be given by Mark Borkum. Mark is a computer scientist at the Pacific Northwest National Laboratory here in Richland, Washington, and he works on the application of computer science and software engineering techniques to enable and support interdisciplinary research. And I've worked with Mark in the context of some work he did at EMSL. And one of the things he has done in EMSL is develop some tools for FluxOmics modeling, which he's going to describe in part today. I'd like to note that Mark is also involved in the Microbiome Data Collaborative, the National Microbiome Data Collaborative, and has done a variety of different types of applications across a diverse set of scientific application spaces. So Mark, are you ready to go?
I hope so. Hello, everybody. Could someone please confirm that they can see the title slide?
I can see your slide and it's in full screen and looks great. Thanks, Mark.
Fantastic. All right, let's get going. So hello, my name is Mark Borkum and I'm a computer scientist at PNNL, and I'm based in the beautiful Washington State in the Pacific Northwest. And you can see an aerial view of PNNL on the title slide. As Tim mentioned, I'm the computer scientist and I work with the domain experts. And this is one of the really cool things about PNNL. We're a matrixed organization. So when you do a project, you work with different people and everyone brings different things to the project. I would say, please direct your questions if they are biology or biochemistry focused to the other panelists. But if they are computer science focused, then please by all means direct them to me. And before I get going, just a quick acknowledgement. Thanks to Tim and Nancy, the organizers, great job. And thanks to Hyun, Chris, and Janica for day 3 today. I think you're really going to enjoy the narratives that you'll be using this afternoon. So as we mentioned, the quick overview, this is an introductory talk. It's not intended to be a graduate level talk. This is to whet your appetite and to try and entice you to come and work with us at PNNL and at EMSL. We're going to very, very briefly look at flux balance analysis, but I'm going to focus on metabolic flux analysis. And specifically, we're going to look at stable isotope labeling experiments and how we might incorporate that experimental data into our models. So what is metabolic network modeling? You've heard it a lot this week, I'm sure. The goal here is we want to correlate the genome with the molecular physiology. We want to know, can we model the behavior that we observe? And can we make our models really, really good? So what is the model? It's a mathematical representation of the genome. Hyun went into a lot of detail there. We're going to focus on the biochemical reaction network, but as Hyun mentioned, there are a lot of other places that you might want to look at for your model. And we're going to specifically be looking at constraint optimization problems. And for example, you might be looking at maximizing an objective function. And as Hyun mentioned, the standard is to maximize biomass production. And for this talk, we'll be looking at biomass generated by the central carbon metabolism. A little bit of terminology here. If I use the word biological entity, I'm referring to a cell or an organelle or maybe some fluid. When I'm talking about the metabolome, that's the set of small molecules, so the metabolites. And when we're talking about the fluxome, we're talking about the set of all of the rates of the metabolic reactions within that entity. And you'll see some graphics in subsequent slides, and they're pretty cartoony, but they all follow the same theme. What we've got at the bottom of the slide, there are some compartments. So we've got an intracellular compartment with some intracellular metabolites, and we've also got an extracellular compartment with our substrates and our products. And you can see that we have flux, the rate of change of the concentrations of those metabolites, and the amount of flux is being indicated by the thickness of the arrow in this cartoon. So here's our narrative. We're going to do a fictional experiment. We want to study the carbon chemistry of the TCA cycle in some strain of E. coli. And in our imaginary lab, we've got a bioreactor, and a little image there on the right-hand side. Our protocol is we're going to bring the sample up to metabolic steady state. And then we're going to control the substrate uptake rate, we're going to measure it, and we're also going to measure the products that are generated. So how are we going to do that? We're going to model the biochemical reaction network. Here is an example of the reaction network for the TCA cycle. And I've taken this from Antonowicz's paper, and you'll find all the references at the end of the slide deck. And yes, we will be posting the slides as soon as possible. The reason that I'm using this example is you can go to the paper and follow through all of the math that I'm going to allude to in the rest of the talk. So what is a biochemical reaction network from the perspective of a computer scientist? It's a graph. It's a labeled graph. So the edges, which correspond to reactions, and the vertices, which correspond to the molecular species, they're labeled with information. In this case, the names or the identities. Edges are directed, and the graph as a whole is cyclic. There is no restriction on which edges can point to, go from which source vertex to which target vertex. You might want to have, in the case of this example, a reversible reaction being modeled by two directed reactions, and in which case we have a cycle. So you can see on the right-hand side, fumarate, FUM, and oxaloacetate, OAC, in the figure. There is V7 and V6, and those are the forwards and backwards directions for this reversible reaction. The other reactions being irreversible in this case. So how do we actually go about writing this stuff down? People tend to write them as reactions in an interesting syntax. And the nice thing about the syntax is we've got these two kinds of variables. There are stoichiometric coefficients. And so these are the things that tell us this many moles of this thing plus this many moles of that thing gives us this much of that thing. And we can balance on that or do other mathematical techniques, as we'll allude to later. And importantly, the stoichiometric coefficients are additive. You add them together. But the reaction coefficients, these rate variables, they're multiplicative. And so in our code, we need to distinguish between them. And this is very important. Let's look at this represented as a matrix. So if you have a graph, you can represent it as an adjacency matrix. And so here we have the rows are the metabolites and the columns are the rates. And you can see that we have, for a specific reaction, if you have a substrate, it's got a negative stoichiometric coefficient in this matrix. And if you have a product, it's got a positive stoichiometric coefficient. And so here we've got alpha ketoglutarate going to succinate and a bit of carbon dioxide. And you can see in the column I've highlighted, we have a -1 stoichiometric coefficient for AKG, and then carbon dioxide and succinate both have +1 as their coefficients. And these coefficients don't have to be integral, they can be fractions as well. So flux balance analysis, I'm going to go through this very quickly because Kuhn gave a great introduction. In general, if you've got M species and N reactions, you have an M by N matrix. And I'm using the capital N syntax from the literature. And then you can compose your rate equation, which combines that matrix with the rate vector and also the concentration vector. And taking the rates of change with respect to the time variable t, we get the flux variable vector. And as Hyun mentioned, at metabolic steady state or at quasi-steady state, depending on your terminology, we're equating all of that to be 0. So we're saying that the rates of change of the intracellular metabolites is 0. And then we use constraint optimization, in this case, linear programming. And we might constrain that, as Hyun alluded to biomass generation. But there are questions here. What can we constrain? What should we do? What must we constrain? And so this is alluding to rational experiment design. And then how do you actually test that your sample is at metabolic steady state? And as the computer scientist, I'm not going to tell you how you do that. I'm just going to say this is a question that you must consider, because the code is always deterministic. It receives numbers and it calculates the answer. So you need to make sure that the numbers you're putting in as inputs are valid. So how do we analyze the reaction network and, in advance of actually doing our experiment, glean useful information that would help us to do rational experiment design? One way that you can analyze the network is to take what's called the left null space of the stoichiometry matrix. And in the literature, you'll see this denoted by Ker, K-E-R. And what this does is it gives you an object which contains within it the dynamic invariance as what is known in the literature as a convex basis. So they have non-negative coefficients in linear functionals. What this really gives you is a matrix with columns. And each column is a linear functional of your rates. But as a whole, that dynamic invariance concentration does not change over time. And this is very, very useful because it allows us to identify all of our rate variables, our fluxes, with either transport between compartments, exchange within a compartment, or variables that are determined by these dynamic invariants. And it's very useful to identify these variables in advance because it means we can choose the correct substrate to help us best understand how those variables are playing out in our model. Another important thing to know is whenever you have a forward-backward pair of reversible reactions, you're going to induce one of these dynamic invariants. So every reversible reaction, and by the way, all reactions are in principle reversible, are adding one of these dynamic invariants to your model, which you would then have to solve for. As Hyun mentioned, the geometry of this space is it's a convex polyhedron, which we call the flux cone. And the surface rays of that flux cone characterize the behavior at steady state. In some literature, they call this the extreme pathways. There are lots of different names for these things. And I've given the reference there. So we can take the kernel of our stoichiometry matrix. Oh, no, we can't. We get an error. And the reason that we get an error, it says the stoichiometry matrix must be a full rank. What does that actually mean in terms of the biochemistry that's going on here? So if we look at our reaction network, you can see that there are 2 instances of carbon dioxide that occur in the intracellular compartment. And the matrix isn't full rank because that carbon dioxide, those 2 carbon dioxide things are actually the same thing, but their concentration and their flux is not flowing into a single variable. And so what we're then doing here is we're adding in an extracellular compartment, and we're putting in, you can see we've got ASP_EXT, so aspartate has an extracellular corresponding metabolite, the same thing for acetyl-CoA, and also for our products. And then we're also introducing B1 through B4. And these are additional rate variables, but what this is doing, if you look on the left-hand side now, you can see in the top left quadrant, we have the original stoichiometry matrix, and then in the top right quadrant, we now have the stoichiometry for the exchange and transport from the extracellular to the intracellular compartment and back again. So if we take the top half of this matrix now, oh, I should actually say in this slide, just to quickly say, those variables as well, B1 through 4, these are actual variables that you can measure in your lab. So they correspond directly to your substrate uptake and your product generation rates. For this example, it's a little naive. You wouldn't necessarily be measuring glutamate. You might want to be looking at biomass, but I just thought I'd add the variables and the names of the extracellular metabolites to the figure. Anyways, now, using the top 2 quadrants, we now can take the kernel of our stoichiometry matrix. And if we look on the right-hand side of this equation, we can see, well, we've got 3 dynamic invariants. So if we look at these, let's see what we can see here. So U1, U2, and U3 are our dynamic invariants. They look pretty complicated, or at least U1 and U2 have a lot of non-zero coefficients. That's quite interesting. U3, on the other hand, that's only got 2 non-zero coefficients. So there's something up with that. We're going to investigate that in a second. Let's look at the first row of the matrix on the right-hand side of that equation, V1. So we can see it's a linear combination of our dynamic invariants. And in this case, V1 is exactly equal to U1. And the same for V2. If we go down to the third row, we can see that V3 is equal to U2. But then when we get to V4, we can see that it's actually 1 instance of U1 minus 1 instance of U2. So V4 is a linear functional of our dynamic invariants, but V1, 2, and 3 are not. Now, other things that we can do with this, this matrix on the right-hand side is we can subtract rows from each other, and we can also subtract columns from each other. This is actually really useful. Suppose that we subtract the row for V7 from the row for V6. And remember, these correspond to the forwards and backwards directions of our reversible reaction between oxaloacetate and fumarate. Well, now we can see the net flux for that reaction. And we can see that it's exactly equal to 1 unit of U1 minus 1 unit of U2. So we've already learned something about the behavior of our network. We haven't even had to do an experiment yet. We've just done this from our model. Now, as Hyun mentioned, whether or not our model actually describes reality in an adequate way is something that we then want to validate by experiment. So we go through our right-hand matrix, and we can then characterize all of our original variables according to the dynamic invariants. And you can see that I've done this for the transport variables and the exchange variables. And then B3, V4, V5, and V6, we found to be linear functionals of the dynamic invariants. And as I mentioned before, we can calculate the net rates for our reversible reactions. So the forwards rate minus the backwards rate. And we can see that it is also a linear functional of the dynamic invariants. So now let's put all that on our model graph. This is quite interesting. So we now know we can see U1 and U2. We can determine those by experiment. We know exactly what we're putting into the model as substrate. And then on the bottom right-hand side of the figure, we can see that glutamate, when it's transported, that is apparently equal to U2. So in our experiment, if we're controlling aspartate, if we measure the product generation rate for glutamate, we can test whether or not those two are equal. And when we find that they are equal, then we have another piece of evidence that suggests that we might be at metabolic steady state. So rational design of our experiment is completely informed in this case by the stoichiometry matrix representation of the biochemical reaction network. But this still begs the question, how do we determine this U3? So suppose that I set U1 to equal 100 and U2 to equal 50, just arbitrary numbers here. The fluxes do have units, but as I mentioned, our flux variables are multiplicative. So if you're writing down the dimensional analysis, you are multiplying these values, these scalars, 150 and so on, by the coefficient for the unit. So in principle, because we have a linear equation here, all of these numbers are dimensionless unless you actually multiply them with a unit, millimolar per grams dry weight per hour. So interestingly here, though, you can see if we look towards the middle, oxaloacetate and fumarate, we can see that in the forwards direction, the flux is always whatever dynamic invariant U3 is plus 50. So that's pretty cool. So we've got this, the flux cone that was mentioned before. It looks like in this case, if we set U1 and U2, it's more of a flux valley. And the valley's kind of width is 50 units of flux. And so that's an interesting thing that we've been able to glean just by knowing the substrate uptake rates. We've now found that we only really have to fit one variable. And in fact, what's interesting here is we don't necessarily have to fit that variable in order to get useful results. We can still measure glutamate and carbon dioxide, and I'll give you some examples of experimental techniques that you might want to use. But should we want to know the complete fluxome of this biological entity, then we need to apply a kind of higher level technique in order to determine U3. There are some other considerations as well. And before I go into the topic of metabolic flux analysis, I just want to point out a few things that as a programmer, I have to consider, and these are issues that I would then raise with the domain experts. The choice of objective function is very, very significant. Typically, people choose to maximize biomass formation from the central carbon metabolism. And the way that they do that is they specify a biochemical reaction that involves almost all of the carbon metabolites in their system with, typically they are fractional coefficients. And there can be a lot of substances featured in this reaction. It's very, very complicated. And it's complicated in the sense that it is, the system is sensitive to the stoichiometric coefficients in that equation. Now, the question that you need to ask yourself is, what is the provenance of those stoichiometric coefficients? Where do they come from? So suppose that you're working with E. coli. Which strain of E. coli was used in the experiment from which those stoichiometric coefficients were determined? Are those stoichiometric coefficients actually suitable for the strain of E. coli that you are working with? Now, typically, if you go into GitHub or you see a paper, people will have copied and pasted and copied and pasted and copied and pasted these things. And when you track it all the way back to the source, you find that it's a book by Neidhart from 1987. And the reference to the book is at the end of the slide deck. If you want to read about this in more detail, besides getting the book, which is excellent, there's a great paper by Pramanik from 1997 where they go into the math very, very deeply and explain the relationship between the stoichiometric coefficients that are determined and the resulting activity in your simulated model. So just something to consider when working at the genome scale. Right, so let's take stock. We're about halfway through, so let's look at our narrative. So our goal was to study the central carbon metabolism, looking specifically at the TCA cycle in E. coli. We've got our bioreactor, and we now know what's going in and out of it, and we have variables that we can set so that when we collect that data in our experiment, we can incorporate it into our model runs. And our protocol at the moment is the same, but what we found is that if we limit ourselves to flux balance analysis only, we don't necessarily know the intracellular flux for the reversible reaction between fumarate and oxaloacetate. And if you recall, that's dynamic invariant U3. but we're making progress. We haven't actually done a single experiment in the quote-unquote lab yet, but we've learned a lot about the system just from the model. So let's see how much further we can go. Metabolic flux analysis. The goal here is we're going to label our substrates, and we're going to label the substrates by swapping out specific atoms. We're going to swap those atoms with isotopic variants. And these are variants that hopefully we can detect by experiment. And the reason that we do that is because with an enhanced model, and I'm going to show you what that looks like, we can then track the movements of those isotopic atoms through the reaction network. And if we then do the right experiments and take the right measurements, we can use that to understand the intracellular flux. Now, there are many, many, many techniques that you can use to obtain this data. We're going to focus on mass spectrometry and nuclear magnetic resonance spectroscopy in this talk. And the reason for that is we have wonderful, wonderful instruments and scientists at EMSL for these two instrumentations, and we would love for you to come and work with us. So let's talk a little bit about metabolic flux analysis and nomenclature. We're going to talk about moieties. Now, a moiety is just a set of atoms. We don't care what those atoms are actually for. They might be one carbon atom from carbon dioxide, or it might be a bunch of carbon atoms for something else. But as far as we're concerned, it's just a set of atoms. And importantly, there are n of them. And we can label each atom, or index it to be more formal, from 1 to n. Now, then the next variable, the next kind of hyperparameter, one might even say, in our model is the excess neutron count. So I mentioned that we're going to swap things out with their isotopic variants. When you have, for example, carbon-12, one of its isotopic variants would be carbon-13, and in which case we've added a neutron. So the neutron excess would be +1. And it's important to know what n and m are in advance because it tells us how many possible variables we'll have in our model. Once we've done that, there are combinatorial objects that we can construct. The first one is called the isotopic isomer, or the isotopomer. It's a brilliant name, really rolls off the tongue, and you can see them on the top right-hand side. And these are all of the possible combinations given the isotopic atoms that one could use in this case, the carbon skeleton of a molecule. And so you can see the first one is carbon-12, and subsequently carbon-12. And then the second one is carbon-13, followed by all carbon-12s. The third one is a carbon-12 atom, followed by carbon-13, followed by all carbon-12. And if we run all the way down the list, eventually we get to all carbon-13. Now, what I've also done on the right-hand side is I've written the total number of excess neutrons in that isotopomer. And you can see that some isotopomers have the same number of excess neutrons. And this is very important because what it means is when you take, for example, a mass spectrum and you're looking at the mass to charge ratio, you may see that a single peak is contributed to by more than one isotopomer. And it's important to understand that. The next combinatorial object that we can construct is called a cumulative isotopomer, or a cumomer. And the idea here is that you imagine that there is an any state. There is a labeling state that can represent any possible labeling. In this case, it could be carbon-12, but it could also be carbon-13. This lets you take what are called logical disjunctions. Now, by doing that, you can, in principle, calculate some interesting numbers from your experimental data. What I'm going to show you, however, and I'm not going to go into too much detail here, I'm going to give you a reference instead. What I'm going to show you, though, is that cumomers are actually elements of an even more general combinatorial object. And the last combinatorial object that you see in the literature are these mass isotopomers. As I mentioned, the m plus some number, those things are your mass isotopomers. So we're looking at, give me all of the isotopomers of this moiety where the excess neutron count is the same. Now, what you find with all these combinatorial objects, and there are others, given an example, the number of atoms is another good example, and they are all what's called a monoid, which is a mathematical object from category theory. And what you find is there is a monoid homomorphism between the representation of the moiety and the representation of the isotopic combinatorial object. And again, I'm not going to go into too much detail. I'm going to give you a reference instead. But what's important here is the cardinality of that set is entirely determined by the variables n and m. And that because it's a monoid, there is always an identity element. So for example, if you're used to addition, the identity element would be 0, because if you add 0 to something, you get the original thing that you started with. Similarly, if you're used to regular multiplication of numbers, real numbers, then 1 would be the identity. And so we've got 4 monoids here that I've listed. There are more. They all have an identity element which corresponds to the empty moiety of 0 atoms and the multiplication element. And then there are also additional constraints. One finds that the isotopimer fraction vector, when represented as a vector, it must be stochastic. When one does the same thing for cumulars, because we're taking power sets for various technical reasons, the system must be stochastic, but it must sum to 2n, which is, there are 2n elements of a power set of a set. And so if each vector for each of those elements of the power set must sum to 1, then the total sum for the sum of all the power sets must be 2n. And similarly, the vector for mass isotopimer fractions must also be stochastic. Now, I'm going to show you examples of all of these later on in the talk. You can actually transform between them. And because they're all represented as matrices, there are other matrices that give us transforms between those different spaces. What's interesting, though, is that not all of the transforms are reversible. So let's have a look at some experimental data. Here I'm using T to indicate the any state for cumulars. And so you can see on the right-hand side, we've got 1, so that's the first carbon atom of this moiety. And either we're talking about the any state, or specifically we're talking about carbon-12 or it's carbon-13. And underneath that, you can see I've done the same thing for the moiety of the second atom only. But now we have the moiety of the first and second atoms. That's on the far right-hand side. And so you can see we have all of the combinations for carbon-12 and carbon-13, but we also have these any placeholders. And that's important, and I'll show you why later on. A little bit about notation. You'll see that I'm writing the name of the moiety, in this case A, followed by a slash, followed by the total number of atoms. And then in curly braces next to it, I've written the atom IDs, separated by commas. The reason for that is you might have more than 9 atoms, in which case you don't want to write the shorthand, because you would not be able to distinguish 10 from 1 and 0. The shorthand, though, is quite useful because we've got limited space on some slides, so I will be using it as well. Note the power set of atoms. So if we have 2 carbon atoms, the power set contains the empty set, and then the set of 1, the set of atom 2, and the set that contains atoms 1 and 2. Now, in principle, if we had all of these different moieties and we put it into, let's just say for the sake of argument, a GCMS, we might be able to measure the mass fractions. Now, the m plus 0, so if we have an empty moiety, the m plus 0, so we're adding no excess neutrons to a moiety of 0 atoms, well, the probability of finding that is always going to be 1. So we'll just have a single peak there. But when we look at A2, and then in brackets 1, so the first atom of the A moiety, you can see that its m plus 0 fraction, the light blue in this figure, is coming from the carbon-12 atom, and then the m plus 1 is coming from the carbon-13 atom. And similarly for the second carbon atom of moiety A. But then when we're looking at the combination of 1 and 2, for M0, that's yellow, we can see that that is getting its kind of value, its intensity, from carbon-12 and carbon-12. And for M plus 2, we can see at the very bottom right of the figure, it's carbon-13 and carbon-13. But for M plus 1, we have to take the sum of the intensity when we have carbon-13 as the first atom, but carbon-12 as the second. And then vice versa, we also have to include the intensity when the first carbon atom is carbon-12, and it's the second carbon atom that is labeled. Now, you can see that this only becomes more complicated as you increase the number of atoms in your moiety, which is why you want to have software do this for you. Now, let's look at other experimental data. Suppose that we want to do this with nuclear magnetic resonance. There is a nice result. Essentially, you're applying Bayes' rule. And what it lets you do for certain types of high-dimensional experiments, one can write expressions, typically they are ratios, and those are ratios of the isotoper fractions that one obtains, and one fits to those ratios. And there is a technique for doing this. I've got an example here for a 2-deoxy HSQC experiment for glutamate. And what you're doing is observing one atom and detecting another atom, carbon in this case, but we are not limited to homonuclear isotopic labeling experiments. One could do heteronuclear experiments as well, for example, carbon-nitrogen. And once we've done that, we get an expression for our observe and detect atoms, and we combine that according to Bayes' rule, and we get, as a result, an expression for the intensity that we expect for that peak. And if you look at the bottom of the screen, you can see I've got the glutamate drawn there, and I've labeled the atoms according to the IUPAC numbering convention. And you'll see that the first carbon atom labeled 1 is on the right-hand side. That's because the IUPAC convention is to start at the carboxyl carbon. And on the right-hand side of the screen, you'll also see I've just got a little bit about the ToxyHSQC. This is quite interesting because the observe and detect maps very, very nicely to the experimental protocol. Right, so we now know that we've got this kind of higher level combinatorial object space. We can measure things in principle with mass spec and with NMR. How do we actually know where things are and then what to measure? And I'm going to cover this hopefully in 10 minutes. So we started originally with stoichiometric coefficients relating all of our moieties, or at least in that case, they were molecular species. Now we're talking about individual moieties, and we're tracking the atoms individually. So all our stoichiometric coefficients are 1, at least for the isotopically labeled things in our model. Now, what I'm doing here is I'm using a shorthand. A, B, C, D is shorthand for curly braces, A, B, C, etc. And the reason I'm doing this is to save a little bit of space. You can see if we look at what each of these atom transition reactions, what they're telling us now is we have a particular moiety. It consists of some isotopic atoms, and those atoms start on the left-hand side and they transition to the right-hand side. And if you look at V1, the first row in this table, you can see that we're starting with oxaloacetate and acetyl-CoA, and the ordering of those atoms in citrate is not necessarily A, B, C, D, E, F. It's D, C, B, F, E, A in this case. To make things even more complicated, you may have moieties that have stereochemistry. For example, succinate and fumarate, they have rotational isomers. Now, we can account for that by having a probability associated with each ordering of atoms. So if you look at V5, you'll see that succinate, there's a 50% chance of observing A, B, C, D, and a 50% chance of observing D, C, D, A. And similarly for fumarate. Now, this is very, very important when you are attempting to incorporate NMR measurements into your models. I should also say the mixing probabilities, they must sum to 1. They don't have to be 50% or 100%. They can be arbitrary positive probabilities as long as the total is stochastic. So here's a particular example. We have alpha-ketoglutarate, or glutamate even, and it's going to succinate and carbon dioxide. And you can see because we've labeled our atoms, we can track where those atoms go. And here's just an example of how we compute all of the atom transitions for things that involve mixing probabilities. So we're starting with the original syntax, and then what we do is we're distributing. So we're taking, it's just like when you multiply out, when you've got things inside of parentheses, you're multiplying each thing with every other thing. It's a Cartesian product. When we take that Cartesian product, we are multiplying our probabilities. And so you can see we start on the left-hand side. There are 4 possible ways of combining 2 things on the left with 2 things on the right. And if we take our atom labels, in this case A, B, C, D, and we index them, so we just count 1, 2, 3, 4, and we track them that way, we can see that the first and the last possible combinations, they are actually identical, and the second and third are also identical. This means that we can sum those mixing probabilities. So we started with a fairly complicated biochemical reaction where the probabilities were inside of the description of the atom labelings. And now we've moved that probability out onto the flux variable. So what we've done is quite literally distributed two functors, to use the category theory parlance. So you go ahead and you do this for your entire reaction network, and here's the result. And you can see we've got a lot more reactions than we started with. We started with 8, and now we have 11. And in fact, once we add in our extracellular compartment, we're going to have even more reactions. This isn't a problem in practice now. So now we can start thinking about rational experiment design. What I've done here is I've plotted as a graph all of the isotopic atoms in the model, and I've related them by the flux variables as shown in the stoichiometry matrix. And so you can see if we're starting, for example, let's start on the left-hand side, we have aspartate 4, and it's the 4th atom, and we can trace it down by V8, and it goes to oxaloacetate, again, the 4th atom. And then things get a little bit messy. But this is actually really, really useful because we can do rational design. We can ask the question, which atoms of the substrate should be labeled? And we can ask that question by looking at which atoms of the intracellular metabolites will subsequently be labeled. And that might motivate the instrumentation that we choose. Here's another way of looking at that information. What I've done here is I've grouped the atoms together by their moiety. And I'm showing exactly the same edges, but this is a little bit more clear for situations where you genuinely want to see the behavior in the context of the moiety as a whole and not for the atoms individually. So now we can ask the question, okay, let's suppose we're going to go to the shop and buy some acetyl-CoA. Which labelings of acetyl-CoA should I purchase? And this is a very important question because budgetary constraints are really quite important when you're planning an experiment. You want to make sure you're buying the right labelings, but also you want to make sure that you're able to purchase those labelings. Perhaps you don't need 100% uniform carbon-13 labeled, to 6 degrees of precision, 99.99999% labeled. Maybe you can't afford it. So let's just ask the question through elementary graph theory, in this case, reachability analysis. If I label atom 1 of acetyl-CoA, or if I label atom 2, which isotopic atoms are subsequently labeled in my model? And you can see that the results are dramatically different. So if we only label the first carbon of acetyl-CoA, we find that carbons 2, 3, and 4 of citrate will never receive any labeling. And similarly for glutamate, if we track all the way through, we can see that 2, 3, and 4 of glutamate will never receive any labeling. But on the right-hand side, if we purchase C2 labeled acetyl-CoA, well, now we can actually detect labeling at carbons 2, 3, and 4 of glutamate. And in fact, if you were designing an experiment, you may want to mix your labeling. That will give you a really complex result that will really, really help you to fit the data to your model, or the model to your data, I should say. Right, so here's our narrative so far. Another quick recap. So our goal was to study the TCA cycle, and now we're using carbon-13 labeled substrate. We're going to bring our sample up to isotopic steady state as well. And we're going to control our labeling and measure the labeled output. And we're also going to take samples, perhaps of the exudate, and we're going to analyze them using mass spec and NMR. How do we do that? We do that with a technique called elementary metabolite units. Again, I'm not going to go into too much detail. I'm going to give you a reference instead. The idea here is we take our atom transition reaction network, we represent it as a graph, and we do more graph theory on that graph. And the graph theory that we're actually doing is we are merging lots of smaller graphs into a bigger one. We're looking for vertices that are reachable with respect to other vertices. So we're looking for sinks in our graph, which correspond to products, where the labeled isotopic atoms can flow all the way from the sources, the substrates in our product. And then we're reducing the number of variables further using a one-in, one-out edges counting approach. All of this is in the Antonowicz paper that is referenced here. The output of all of that is a system of linear equations that one does constraint optimization upon. So you're minimizing the error with respect to a cascade system. So one goes ahead and does this. Here is the example for alpha-ketoglutamate glutamate. And what you have are reactions that involve a given number of isotopic atoms. In the literature, they refer to this as EMU size. So we have 5 isotopic atoms in this case, and so there will be EMU reactions from sizes 1 to 5. And you can see for size 1, we're tracking the individual atom transitions, but for size 2, we're now tracking 2 isotopic atoms at a time, then 3, then 4, then 5, then N, and so on. So we do all of this, and again, we can plot all of this as a graph, but now our vertices are these moieties, and moieties of moieties, and combinations of moieties, and so on. But we can plot it as a graph and do some more experiment design. So let's see what happens if we label acetyl-CoA's second carbon atom. Well, now we can see how that labeling is flowing through all of our moieties. If we do this contraction operation that I mentioned, you can see that the graph is a little bit smaller, which is great because the adjacency matrix representation of this graph will have less rows and columns, which means that any mathematical operations that we choose to do will be less computationally intensive. Again, let's label that carbon atom, and we can see how the isotopic label is traveling through the network. So EMUs, just to kind of recap here, we're still at quasi-steady state. We're still doing all of this with respect to an objective function, as we would have done with FBA, but we're now also adding constraints to minimize the error from our measured value against expressions of these combinatorial objects. So just to finish up the talk, so now what we have, the experimental data. I mentioned that you might want to vary your labeling. So here we have 50% of natural abundance, but then we're using 50% C, or 25%, sorry, C2 labeled and 25% uniform labeled. And our aspartate is completely natural abundance. And then we might go ahead and do some gas chromatography mass spec. And here I've done the headspace gas. Again, this is all fictional. So we've got the mass isotopomer fraction vector for carbon dioxide, the extracellular carbon dioxide. And then we also have GCMS of glutamate. And we have GCMS of our intracellular metabolites. Now, notice that we've been able to, in our fictional experiment, associate our peaks with the moieties of the molecular species. So for example, the first row is fumarate, has 4 carbon atoms in total, but we're only considering the moiety of 1 of those carbon atoms. Now, whether or not this is reflective of reality, we can debate over a drink, but this is an example of experimental data and how one might incorporate it into a model. Next, we might do some NMR experimental data. So I mentioned we might take the exudate. So here I've done this fictional ToxCHSQC on the extracellular metabolite glutamate, and we've got a bunch of numbers here. These would come from the NMR spectra. We put all of that in, and lo and behold, the number is 75. This is good because it reflects the number that you would find in the original figure in the paper. So what we've done now is, starting with just a description of the biochemical reaction network, we went as far as we could with flux balance analysis, and we decided to apply metabolic flux analysis with a rational experiment design. And as you saw, MFA allows us to elucidate both the intracellular and extracellular metabolic fluxes. So just to wrap up here, I mentioned please come and join us at EMSL. You'll find the link there in the bottom corner. One thing that they might not have told you is we do have the guest house. So when you come and work with us, there's a place where you can stay. The interesting thing about FluxOmics, EMSL is currently defining itself based on science areas that we are really interested in. FluxOmics spans both of those science areas. So if you want to come and work with us to do FluxOmics, your proposal has a really good chance of being selected. And then the last thing on the right-hand side, you can see the user facility has a whole suite of capabilities. And with that, I will say thank you very much.
Thank you, Mark. That was amazing. Actually, I really learned a lot from your presentation, so I appreciate that. And being able to add to flux balance analysis and other methods the ability to directly measure some of these fluxes through isotopic labeling and metabolic flux analysis is super powerful. So thanks very much for the talk. One of the things we're getting from, I think one of the questions that came through is that the talk was super. People really enjoyed it, but there's a huge amount of information presented especially for people for who this might be their first exposure to metabolic flux analysis. So I'm wondering if you might provide, and maybe the easiest way to do this is to go over to the Discord channel and provide maybe a key textbook or one or two key links that people could follow up to. I know you're going to provide, a more detailed list of references, but if you could give that to the people, that might be something that would be really helpful. And then one of the things that I was going to ask, and this just came up in the Zoom chat also, is are these tools publicly available? And in particular, is there any relationship to KBase, or either current or future?
That's a really great question. Just to address the first comment, yes, I will put, I'm looking at the books right now, so I'll put the citations for those into the metabolic modeling channel.
Great.
And then the tools. So the answer at the moment is yes and no. FluxOmics at PNNL has a very long and deep relationship with KBase. And we've been talking for a long time about putting metabolic flux analysis tooling into the KBase. The question at the moment to be discussed, well, there are two of them. The first one is, to what extent do we put the modeling tooling into KBase versus the visualization and rational experiment design tooling? And then the other question is, who pays for the work? We have a lot of codes that we are in the process of open sourcing. And once that is done, there is a nominal step to wrap those codes up as KBase apps. So once we've open sourced our codes, we would probably go through PNNL's internal proposal system to get that funding, but we would also be very open to an open source project if someone would like to work with us to wrap that code up as an app on our behalf.
Great. Excellent. We're a little over time, but I'm going to hit one more question from the Zoom chat and then some other questions. Mark, if you want to head over to Discord and chat on some of the other questions that have come up. But what are considerations around isotopic fractionation in terms of FluxOmics?
So that's definitely one for the experimentalists. From a computer scientist standpoint, which is really the only valid answer I can give you, the most important thing in your code is to be able to identify, and by identify I mean be able to read and set a value of a variable. And so when I talk about these combinatorial objects, the code that I write, my software, has to be able to identify all of those different combinatorial objects and all of their things that are within them and the associated variables. That way, whatever the experimentalists do come up with, we can at least attempt to accommodate in the code. But I can't answer that question from an experimentalist standpoint.